Thank you, Alexandros. Very nice introduction, and it's really a pleasure to be here. I hope that we will have a very nice round of discussion afterwards. And yeah, let's start. As Alexandros mentioned, I come from Italy, and I come from an island that is called Sicily in the south of Italy, which is probably famous for the mountain Etna, which is the highest active volcano in Europe. And there are some sites also nice, like this is in Agrigento, which next year will be the capital of culture of Italy. So if it happens for any of you to come visiting, I think it's a good chance to look at it and look at the island a little bit. However, despite these beautiful touristic attractions, there are also a lot of refineries, industries, sites, and pollution. So just next to those places that I showed you before, there are like these industries where they produce a lot of pollutants released in the environment. And it has been shown in the WHO report that in those areas where these pollutants are released, there is also a higher incidence of several diseases such as cancer, different type of cancer, leukemia, and so on. So this is just an anecdote, but I will come back to it at the end of this talk, both because I'm close to this topic and because I come from there, and also I want to show how this tool that I'm going to present today can be helpful to understand the impact of environmental exposures on a molecular basis. And the agenda of today will be grouped in 3 different parts. The first part, we will look at the theoretical aspects of this work, how we try to connect chemical exposures to molecular genetic perturbations. There is a second part instead, which has a little bit more of biology and experimental setups. And there are 2 QR codes here. You can try to scan it now. You can scan it later if you don't manage right now. This just linked to 2 papers that I will talk through this talk. And the last part is connecting to environmental perturbation worldwide. And then I will focus a little bit in Sicily once again, so that I can connect to the beginning. But for now, let's focus on the first part. And when I talk about environmental exposures, I mean everything that is released in the environment. That can come from medications, but also industries, as we have seen before, habits like smoking, diet, and so on.
Sorry for interrupting you.
Yeah.
There is a message in the chat saying that the only slide is visible. Is the same problem for everyone online, or can other people verify that?
I can go back.
No, it seems like one person was the problem. Okay, sorry for interrupting again. But
no problem.
To clarify this.
If there are burning questions, or if there are other problems, please interrupt me anytime. Really, no issue with that. So these chemical exposures, they are everywhere, really. And nowadays, luckily, we put some computational effort to annotate each of these chemical compounds and chemical exposures in large database. And the complexity is huge. Like the possible chemical structures that are in some of these databases are over the billions of data. And we have used one of these databases, the comparative toxicogenomics database, which put together chemical structure, chemical information, and genetic information of genetic perturbations to then create a framework that could detect the role in biology, in human biology, of these chemical compounds. Just to give you a brief idea of this database, how it is structured, it contains millions of chemical genetic interactions. And they are, on the first round, they are extrapolated by text mining. And so afterwards, they are curated by experts in the field so that they provide a certain level of confidence for these chemical gene interactions. They have a lot of other informations as well, such as anatomical size, organism, and so on. But what we have used in this study was just the chemical gene interactions in humans. And the way we have done that is that you will see in a minute, we have created a bipartite graph that I don't want to spoiler it because it will be just in the next slides, but that allows us to connect the chemical to the genome. Just to have a brief overview on the statistics of this comparative toxicogenomics database, CTD in brief, we can understand that most chemical compounds are organic chemicals, which is in line with the knowledge that we have of the world where we live in, because organic chemicals are chemical compounds that have at least one carbon atom. And because we live in a carbon world, then that makes sense that the majority of these chemical compounds are represented by this category. The second grouping that we can do is based on the type of interactions. And we can observe that most of these interactions are change in expression. And what I refer to it is that these are changes in mRNA expression, so transcriptomic changes. And that is most likely because in the last decades, there have been a huge effort on using this transcriptomic readout as an experimental readout. And while there have been less exploration on mutagenesis or protein stability and so on. So this could lead to some literature bias, and we have tried to address this on our computation. The way we have done that, as I mentioned before, is creating this bipartite graph. So a bipartite graph is a graph where in one side we have a group of nodes that share a typology, that they are in the same group. In this case, those are exposures, so chemical exposures. On the other side of the graph, we would have another type of nodes, which are genes, and then a connection is built whether there is an association or not. And once we have a bipartite graph, we can project that in two sides so that we will obtain a normal network where each node is an exposure. And an age is built whether they share a significant amount of genes that are perturbed. And on the other side, we have a gene-gene network. So each node will be a gene, and an edge is mapped whether they have a significant amount of exposures that they perturb the same genes. And you might see that the width of this edge change, and that change based on a certain weight that we have defined. And specifically, when we map these two networks, we would observe that they are very, very dense. So we need a way to sparsify it. And the way how to sparsify it was, first of all, to compute a p-value for each pairwise interaction, pairwise of genes or exposures. So through an hypergeometric test with multiple correction, we were able to distinguish which were the meaningful one. But on top of that, we have set a weight that was a Jacquard index on in each of these edges, which will compute the amount of shared component between two exposures or two genes. And we have further used this weight to use a disparity filter approach so that we could sparsify this network. And I'm not going too much in the technical details here, but if there are questions later on, we can go more on the techniques that have been used through this work. Well, what you should know is that in the beginning they were fully dense network, and they became sparse in a way that you can even plot it in 2D, and you will see something like this. So this will be the exposure network on one side and the gene network on the other side. and they provide really complementary information. So they are like two sides of the same coin. And they contain information for almost the entire genome, covering a bit more than 500,000 of interactions. And what we can observe from these networks is that we can analyze it in two different ways. One way that is looking at the global topology of these networks, and another way instead is the local topology. In the global aspects of this network, we observe that if we look at their degree distribution, both of them, they follow a certain shape that is similar to a power law degree distribution, which means that we have the classical shape of the rich get richer, as in social network. But when we look at the rich, in this case, in the exposure networks or the hubs, then when we zoom on them, we would observe that most of these exposures are fairly unknown exposures, and you can see also the number of perturbed genes for each of them, which is not a lot, which we believe that is able to contrast for the literature bias, because when we look at those few genes that are perturbed by these exposures, those are actually very fundamental genes in the cell, in the human cell for survival, because they are either caspases or members of the BCL families, or are responsible for the apoptosis pathway, which means that it's likely that these exposures were understudied, and if they perturb one of these genes, it's very likely that they will perturb way more genes, impacting the cell survival and the cell functioning. On the other side of this bipartite graph, there was the gene-gene network, and also this gene-gene network follows a sort of fat-tail degree distribution, where we have very few genes that are well connected in this network. And when we zoom on these up genes, they are actually fairly unknown genes. Some of them control long non-coding processes. Some of them are have very few biological functions associated with. But what they have in common is that they are perturbed by a broad class of exposures, and some of these exposures are actually among the most perturbing exposures in CTD, such as benzopyrene, valproic acid, and aflatoxin B1, so that they cause a broad perturbation in cell survival, causing more than 10,000 different genes that are perturbed by them, which again speaks for a better characterizations of these genes that most likely were underappreciated before, but that instead are also taking part to fundamental processes in the cell. That was for the global aspect of these two networks. But a second step that we can do is looking at the local structures. So when we have a network like that, where we have exposures and nodes and ages that correspond to the shared genetic component, looking at the local structure could mean also defining communities. And so then we can run a network community identification algorithm as your choice. In our workflow, we have run two different algorithms because they were quite different in their method. One was Louvain and the other one was InfoMap. And in both ways, we have retrieved something interesting, that this community classification or chemical community classification was actually a hierarchical community, which means that within a community, we could identify a subcommunity and again as another subcommunity and so on. And then you will see in this tree map here, and that would be explained in a minute with an example, with just an anecdotal example, but I think it's clearer to understand it. So when we look at this network, we can zoom in one of these community, for example, community 10, and we call it as a broad community. And within this community, we can identify different subcommunities based on the modularity of these exposures, how they are connected. And again, we can zoom in one of these subcommunity and identify an intermediate level, and then again a fine-grained level. And by looking at this C1022, this community here for exposures, we can observe that these 4 exposures are fairly different chemically. They are very different. Although you might have some feeling that a bell is ringing, because you have a calcium heparin here and a dalteparin, which are 2 molecules of the same family of the heparin. But besides that, they are fairly different, these chemical exposures. However, all of them target a specific protein, which is called SERPINK1, that is responsible for the production of antithrombin. And that means that all of these chemical exposures indeed cause a perturbation of the term of complement deconchylation cascade. And then if we dig into the nature of these molecules, we know that calcium heparin and dalteparin are used actually in the clinical practice against thrombotic events. So we know that this association makes sense. Then when we look at oxalic acid, it's a little bit less trivial, the association, because this is a contrastum medium which is used when you take an examination such as a CT scan. However, it has been shown that as a side effect, it could cause thrombotic events. And for that reason, in case the patient is susceptible to thrombotic event, then he should use a different contrast medium. And finally, clobazam is an antiepileptic drug, which again recently has been shown to have as a side effect the possibility to cause thrombotic events. And similar to the oxalic acid, should be considered to use a different drug in susceptible patients. So this was just to say that it seems that the fine-grained communities are able to capture some similar biological activity. And this is not so unexpected, because we build these networks based on the shared genetic perturbation. Now we wanted to test whether that was the case for all communities. So we had taken all the communities, whether they were broad, intermediate, or fine-grained, and we have plotted here their, let's say, biological similarity, which was basically a genetic overlap calculated through Jacquard index, against the community density, so network density within each of this community. And what we found out is that there is really a nice direct linear positive correlation of network density and biological similarity, which means that when we look at the higher, very dense connected modules or community in these networks, these tend to be small community or belonging to fine grain, so fine grain communities, and they also tend to have a higher genetic overlap, so that they tend to perturb the same genes, or chemical exposures belonging to the same community tend to perturb the same genes. The next step that we wanted to check was, when this chemical exposure perturb different genes, what do they do? Would they converge to similar pathway or not? So we have then perform an enrichment of these genes for different ontologies, such as the gene ontology or KEK pathway. And the next step was to quantify, let's say, the similarity of enrichment terms or enriched terms for each of these chemical exposures. And we quantified with a measure that we define as biological cohesion. And this is basically the Z-score of the largest connected component of the rich terms in the gene ontology tree for the biological processes, molecular function, and cellular component against 1,000 random sets of the same size of enriched terms. And what we found out is that the highest z-score would be found in the fine-grained communities. So which means that at the fine-grained level, the smaller community that are very dense, those are also the ones where the enriched terms, enriched biological terms, tend to be also more cohesive, more similar, in the same part of the gene ontology graph. So this is a very simple way to quantify biological cohesion or biological similarity among terms. The next step was actually to make use of the gene ontology, which is a hierarchical ontology, as you might see here. And when we highlight two terms, such as, for example, two terms of interest like that, we can quantify their connectivity either based on their tree that connect them, or instead we can make use of another measure, which is semantic similarity. And the semantic similarity makes use most of the times to a term that is the common ancestor, or the most informative common ancestor, which is this one here. And then that term is used to calculate the information content, which is let's say a probability function, or an information that is found for each term in an ontology, in an hierarchical ontology. And that allows us to calculate a value that is called Resnick. And this Resnick value is basically a semantic similarity measure, where higher value shows that there is a higher similarity between two terms, and lower value is lower that similarity. We can run that through all pairways of enriched terms. And what we found out is that the median Resnick value in the gene ontology was always higher in the fine grain compared to intermediate or broad communities, which means that also based on the semantic similarity, it is confirmed that chemicals tend to perturb genes that have a certain biological activity that is kind of similar. So all of this was to try to show that grouping exposures accordingly to their network density in this exposure network allows us to capture a group of exposures that have similar biological activity. However, there is another question that arises. Are these group of exposures also sharing a chemical similarity? Because now we show that they have a biological similarity. Do they have also a chemical similarity? And to answer this question, we use still the Resnick score, but calculating on a different ontology. And this ontology was a chemical ontology, where each node would be a chemical compound. And we can map these chemical exposures that are in the exposure network on this ontology, calculating the average pairwise Resnick within a exposure community, and the comparison between exposure communities. So then I will compute all the pairways of exposures between communities. And what we found out was that the distribution of Resnick's values was always higher within a community compared to between communities, which means that the exposures that belong to the same exposure network community tend to be also chemically more similar to what you would expect if you would randomly pick them in the entire network. And on top of that, we also observed that that was even more true when these communities were in the fine grain level. So these communities were smaller, and they were even more dense. So that meant for us that the fine grain communities were able to recapitulate both the chemical similarity and the biological similarity. And we can actually show that also with a nice trend or correlation when we group the chemical exposures belonging to the same community based on their genetic overlap, so their Jacquard index on their biological similarity, and we plot that against their chemical Resnick similarity. And we observed that those exposures that have the highest genetic overlap, so those that are more similar genetically based on their target genes, they tend also to be more similar chemically. Well, whenever I said chemically here, I meant again calculated through the Resnick similarity, which is a semantic similarity on the chemical ontology. However, we observe and we show also that this chemical Resnick similarity correlates well also another type of similarity, which is called Tanimoto similarity, which is a structural similarity used by chemical people to quantify how two molecules would be similar from a chemical fingerprint. And that would tell us that indeed there is a correlation between structural chemical similarity and biological overlap. And all of this was to say that these groupings, this community defined on the exposure network, are useful because they are useful to group together exposures that belong to kind of the same category based on their biological activity. However, if we want to further explore their biological activity, we decide to take another step, which was to make use of the protein-protein interaction network. And this is a network where each node is a protein, and an edge is built whether these nodes are in physical contact among each other. So if two proteins interact physically, then they will share an edge. And when we map the proteins that are perturbed by those chemical exposures in the protein-protein interaction network, we observe that we can classify exposures in three different categories. We have those exposures that target AB exposures, or that target AB genes, and we call them as AB exposures. And AB genes in the PPI means to be genes that are connected to a lot of other proteins. Or in this case, I'm using the term genes or proteins interchangeably, but I'm referring to proteins. Then we would have the peripheral module exposures, and those are exposures that target group of proteins that are in the periphery of this network. So they are well connected among themselves, but they are not so central, so not so connected to the other proteins in the network. And finally, we would have central module exposures, which are exposures that target central modules, which means proteins that are well connected and are also central in this protein-protein interaction network. And we show that this classification is not just a topological classification, but has also a repercussion on their biological activity. So first of all, we can observe that most exposures in the CTD are actually targeting one hub in the protein-protein interaction network, because around 60% are either central module exposure or hub exposures. And we can quantify that also by mapping a certain random threshold of significance, and showing that in most cases we have let's say a Z-score that I don't want to go too much in the details, but is a measure of significance on of an exposure to target a specific protein that is central in the protein-protein interaction network. But what is instead, I think, more meaningful is that once we have this grouping, so we have these categories, we can actually map to their biological activity and their hazard. And with hazard, we mean like how much an exposure is deleterious for a cell or a human cell. And what we can observe is that central module exposures, which are those that are connected and they are central in the PPI, are the most harmful one, followed by the hub exposure. So it seems there is almost a correlation between nociveness and centrality in the PPI, And we wonder whether we could test that, and we could do that. So what we observed, it was indeed this trend, this positive trend between the hazard and the degree on the interactome, on the protein-protein interaction network. And so if an exposure would have at least one up, or would target a well-connected protein in this network, then it would be likely that that exposure will be also harmful for the human cell. What we had done next was to correlate these two diseases. And I will be brief now, but I will go more in the details in a minute, just to explain you the concept that we have a certain chemical X, and this chemical has several proteins that can be mapped in this protein-protein interaction network. And then we have those proteins that are associated with diseases. Then we can say that a chemical and a disease are associated if they are close enough in this network, and they are not associated if they are distant in this network. And I will define what is a good measure of distance later on in this talk. But for now, you just have to know that we have performed this experiment for a lot of exposures, 10,000 almost, and 6,000 diseases, so that in the end we have something like 60 million possible combinations. And among all of these pairwise combinations, we were able to find the most significant ones, so the ones that were more associated to each other and that would relate then to a specific phenotype. And with that, I have finished my first part of the talk, which was the ones that was a little bit more technical, more theoretical. So then I will move on to the biological demonstration. But maybe if you have some burning question right now, we can just stop a minute and then proceed. Otherwise, we can also just proceed and then take all the questions later on, as you wish. What do you think, Alexandros?
Up to now, I don't see any questions in the chat, so maybe you can continue and we can take all the questions in the end.
Cool. Then I follow up with the more biology-driven question, which was related to a chemical compound that is present everywhere. This is bisphenol A, and I'm saying that it's present everywhere because this is a constituent of microplastics. And you might have heard of microplastics a lot, especially in the last years in the media, because they have been found in the cans, in plastic bottles, in toys, and so on. And it has been found also inside us. It was here in Vienna that for the first time it was found in the liver in 2018. And later on it was found also in the blood, and it was found again in the placenta. It was news of today that it was found in the testicle as well. So it's important to investigate the role of this chemical compound, and it is important also to study the development, because later on this can pass through the placenta and it can impact on the embryo. And so we were wondering how this chemical compound will change the cell fate or change the interactions of the proteins that are responsible for a normal human embryo development. And the way how we have done that in collaboration was that we started with human-induced stem cell setup, which are, let's say, cell lines that are made pluripotent again, so they can differentiate to many other cell types. And we decided to go for two different cell types that were cardiomyocytes, so those cells that are constituents of the heart, and neurocytes, those cells that then are responsible for creating the brain. And that came with a cultivative time or a cell culture of 21 days. And during this culture time, these cells were exposed to different concentration of bisphenol A. And all of these concentrations were actually sublethal concentration. So the cell could survive. And these concentrations are similar to the ones that are found in the human environment worldwide. So it is actually relevant and interesting. And what we found out phenotypically was already interesting. So what we found out for the cardiomyocyte was that there was a change in the normal pattern on the frequency behavior of these cells. So these cells would organize themselves and starting beating, as you might see here in this video. And they would beat regularly with a certain frequency and a certain amplitude. However, when they were exposed to bisphenol A, they would beat less frequent and they would beat with way less intensity, as you can also see from the plot here. So there was something phenotypically occurring that was wrong. And that was for the heart. But then in the brain, it wasn't so much better, because when they grew, when they were growing as an organoid, so as a spheroid 3D structure, the structure of the brain organoid in normal setting was always bigger than in the settings where there was bisphenol A. So we wanted to investigate this on a molecular level. And for that, we ran a proteomic analysis on these cell lines, and we identified some differentially abundant proteins. We used then those proteins to map them in the protein-protein interaction network, and we identified a core of specific proteins that were perturbed in the by bisphenol A in this protein networks, in this protein-protein interaction network. And if you are a biologist working with these specific proteins, you might realize already that in the heart, most of the proteins were related to collagens, which is associated with the extracellular matrix, which is important to provide friction to the beating. While instead on the brain, we would have proteins that participate to, let's say, glycolytic pathways, such as enolase 2 or enolase 1, aldolase A, which for the brain is particularly important because the glycolytic pathway, the glucose, is the main energy source for these cells. And with less energy, there is less proliferation, and so there is less growth as well. And we have used these core modules to apply the method that I showed you before of relating these exposures to diseases. So we have collected heart-specific disease and brain-specific diseases. And then we mapped the shortest path that will connect them. And we could observe, let's say, the path to disease, starting from bisphenol A differentially abundant proteins that are in the left side in red, to reach then the disease proteins. And it was pretty interesting because, for example, in the brain, we were able to identify some biomarkers that then have been tested also in post-mortem biopsies for certain diseases, such as amyotrophic lateral sclerosis. And we observed that the downregulation that was occurring caused by bisphenol A was also found in the biopsies of these patients. So of course, we are not saying that bisphenol A is causing those diseases, but for sure is causing a protein signature or protein perturbation that is similar to the one that is found in disease context, and that potentially could lead after a chronic exposure, or at least could contribute to it. And finally, I will talk about the environmental perturbation. And I told you before that we can quantify the relationship between chemical exposures and disease, and I told you that we define a certain distance, and this distance is defined as below here, which is basically the closest distance between two sets of proteins in the interaction network. And these two sets, in this case, are the exposure set and the disease set. In this way, we can quantify this distance, and the smaller is this distance, and the closer are those two sets, so exposures and diseases. And then we can compare against random sets, and we have done that for 1000 times for each comparison, so that we can obtain a z-score. And in this case, a negative z-score shows how close two sets are. A positive z-score instead shows that those two sets are very distant to each other. And what we found that was super interesting for us is that there was a clear correlation between this interacton proximity z-score, which, as I said before, the more negative, the closer the two sets are, and the number of articles. So what we have tested here was to take pairs of chemical exposures and diseases, check for their interacton proximity, and then text mining the scientific literature for the number of articles that would show the co-occurrence of that chemical compound and the disease. And because both the chemical words and the disease words are quite messy, because they use a lot of similar terms and synonyms, we have taken into account also that, so that we could take also synonyms into account. And what we'd observed was that the closest the chemical exposure and the disease were, and the highest was the number of articles that would be retrieved that would talk about the same pairs, chemical disease pairs, which was very interesting for us because it would be a way to validate our measure of interacton proximity. And a further step to validate that was actually to take real data. So what we have done was to look at the incidence of those diseases worldwide that were close to some exposures. So we checked the concentration of the exposures in each country, and then we extrapolate those exposures that were particularly abundant in each of these countries. And then we have taken the diseases that were close to these exposures that were particularly abundant in each of these countries. We have looked for their incidence, and then we have computed a z-score incidence compared to the worldwide incidence distribution. And in this case, the higher this z-score is, the more represented that disease is in that particular country, while the smaller the proximity z-score is, and the closer molecularly, so on a molecular level, the disease and the chemical exposure would be related to each other. So what we would expect in an ideal world was that there would have been a negative correlation, so that more closer would be the exposure and the disease, then the higher the incidence of that disease would be present when that exposure would be around in a particular country. And even though it is not a perfect negative correlation, we could see this trend that is respected. So that when we have a closer relationship on a molecular level, we would observe also an higher diagnosis on the incidence phenotypically worldwide. And as I said before, we have performed this for each country, so that basically we can map each of these countries. And a follow-up was that we could actually take the incidence of these proximal diseases and the incidence of distant diseases. So those diseases that are distant for the exposures that are highly present in each of these countries. And then we will test whether these incidents would have been statistically different or not. So that would have been done just by a normal t-test for each of these countries. And what we observed was that if we consider both air or water exposures, so those chemical compounds that are highly present in the air on water in each of these countries, we would observe that the incidence of those proximal diseases would be always higher compared to the incidence of those distant diseases, which is nice because for us it recapitulates the phenotype, so the incidence, also on a genetic level, so based on just the molecular connections on the interactome. And following up on this, we can actually create a map. We can create a map of Europe in this case because we obtain data for Europe, and this data follow the environmental pollution on the last 25 years. And in this map, we can retrieve a score of risk. So a risk score for each of these countries. And you might see that the worst country is UK, but the second worst is actually Italy. And this risk score takes into account different components. So we have a component that is just a normalization factor, which is the possible.
Salvo, we cannot hear you. You are muted.
Now you can hear me.
Yes.
Sorry. How much did you lose of me talking?
10-20 seconds.
Okay. Then I think we can just discuss about this map, right? This Europe map. Because here we said how we could relate the molecular profile of diseases that were close to environmental exposures in air and water for each country. And what we have found out next was just to take these countries and map a sort of risk quantity of risk disease score based on the exposures that are released in the last 25 years in the environment, either air or water. And what we found out was the UK is actually the country with the highest risk score, so the most dangerous one.
Again, you muted yourself.
That's weird. Now I'm.
Sorry to jump.
Yes.
Do you have your phone or some other Bluetooth item, headset or AirPods or the like connecting? Because that can often steal the microphone.
I am with my headphones. But I don't have anything else connected. So it's through cable. I don't know. It's weird.
I'll go dig in the back to avoid it from happening again.
Okay, thank you. It seems that it's this slide specifically that as soon as I put this slide, then I'm muted. I try to be quick on this slide then. What we wanted to obtain in this slide was just to create a risk score globally in Europe. And this risk score would be quantified by a normalization factor that would have been the highest risk score all over Europe. And then it would take into account the concentrations of the exposures that are released in each of these countries over time, so that we will sum up then for each exposure. And then it will take into account also the degree of the target of the gene that are targeted by these exposures in the interactome, because we show before that there is a clear connection between the degree in the interactome and the harmfulness or the hazard of these chemical exposures. And based on that, we can retrieve this kind of map. And I will just show once again the situation, well, first globally and then for Sicily. Globally, I show you one example for Italy, not just because I am from Italy, but also because it fits well this nice example. This is Endrine, and this is a pesticide that was banned around the 80 because of its toxicity. However, from our analysis, it seems that in Italy there are still high concentration of Endrine in the soil and in the water. And we compare it to France, where it seems that there are low concentration of this pesticide. However, France is similar to Italy for social economic status, also for healthcare system, for welfare, and also for number of chemical exposures that are released in the air and in the water. So for us, France is really the perfect comparison. And what we found out was that if we look at the incidence of those diseases that are proximal to Endrine, so to this pesticide, that are mainly cancers, the incidence in Italy is almost always way higher compared to the Western Europe average incidence, and also to France, up to even fivefold increased incidence compared to France for myelodysplastic diseases or neoplasm. And then we found out actually that, especially in some regions in southern Italy, such as Naples and Puglia, it has been shown that these Endrine or Endrine-related diseases were particularly present in the territory, and that cause highest incidence for cancer access in the hospital in those regions. And finally, to go back to Sicily, as I promised in the beginning, we found out that most of the exposures that have been released in the air by these industrial sites were actually grouped and categorized already, described by WHO. At least there were around 10-ish of them that were described. And they described some diseases that were overly present in the territory, in that region. And here I highlight those diseases that have a unique ICD code, so that they are quite distinguished, and which overrepresentation was present both in the local reference and in the regional reference. So those were diseases that you can be sure that they were more present specifically in that areas. And these are coming from epidemiological studies, so just from the number of access in the hospital. Then when we correlate with with our framework, so with the diseases that are predicted to be closer to these exposures that are released in the environment, then we will find out that most of these diseases would have been actually predicted, and most of them would have been predicted and closer proximal to more than one chemical exposures, and some of them even to many of them, because most of them act in a similar way, causing mutagenesis in the DNA, and then causing multiple cascade events in the cell that then will lead to several cancers and not just one. And with that, I sum up the three parts that I presented today. There was a theoretical part, how we build this framework. Second part, more biologically driven, to investigate the role of a specific chemical exposure which is present in microplastics. And we have done that through the embryo development. And finally, I hope that I convince you that this molecular framework can be used also to identify overrepresentation in the environment, and so that relates nicely to epidemiological findings. And with that, I'm happy to take any questions, and I really thank the organizers for this opportunity and you for the attention. Thank you.
Thanks, Salvo, for this amazing talk. It was very informative and a lot of interesting points for the chemical exposure. If people from the audience have any question, please post it in the Q&A. Okay, if not, I don't see any. I can have one or two questions. First of all, in the part of the on the method, you say that you create a gene-gene interaction network. This network consists of proteins that you use afterwards for the next step, or they're excluded from this part of the study?
Yeah, that's a good point. There is an entire part of the talk that I neglected today, which is regarding the gene network, and we haven't used it for translating on the protein level, but we have used it to reclassify or group genes accordingly to their chemical exposures. So what we found out is that we could identify a group of genes that would be more susceptible to certain exposures, and then that also will translate to certain specific diseases.
Okay. There is a question in the Q&A tab. If you can read it, I can read it briefly. In computing distance between two node sets, you used a kind of average shortest paths and compared to random. Did you consider using other distance measure such as shortest path random walk instead of shortest path random walks?
Yeah. That's a really good point. We haven't implemented other type of distance measures for the randomness. We have kind of agreed, or like we've shown an agreement between the distance of this separation coefficient that we have used and just the average shortest path or the shortest path together. But we haven't compared to random walk, for example, like with visiting probabilities. This is something that, yeah, it could have been done, but yeah, for matter of choices and time, we haven't looked at it.
And something that naturally pop up, how you plan to extend this work, or how do you plan to continue? Because there wasn't any opening or any like the future plan. What's the direction you see on this?
So my dream is to move it also to some policy, a more data-driven policy country decision, because what we observed was that across countries there was a lot of variation on the choice of chemical compounds that were monitored in the territory, in the water or in the air. While in this way we provide really a quantification on which chemical exposures would be more important to monitor instead of others. So my dream scenario is that we could find a sort of agreement or guideline to show which exposures should be theoretically follow up over time in agreement across different countries. And then that would be a new starting point to actually relate to some, let's say, procedure to limiting the disease effects or disease prevention. For example, if we identify that in a certain coast, in a certain area, there is a prevalence of a certain pesticide that would cause an increase of X% of disease incidence in that area, then we could actually point to a different molecule or a different pesticide that could reduce the number of diseases or the harm generally in the population.